Hello, class. Welcome back. Class number 3 or 4 here. We're doing protocol schematics today, and it's a little bit of a dry lecture because of the kind of intricacies of this. But what I really want to do today is stress the importance of what a protocol schematic can do for you. The main thing that it's going to do is take a very complicated protocol and make it easy for a wide audience to understand. So, what are we going to do today? First, define protocol schematics. Then, we're going to talk about why we need them. So, what are the different examples? What are the different kind of venues that you would see a protocol schematic used? How do they differ when you change those different venues? Then we're going to look and make a few examples of protocol schematics. I'm going to go over one, which is a subject recruitment tree, which is a special kind of protocol schematic. And then I'm also going to review how to make a protocol schematic for a presentation. This is something that I find people miss a lot with presentations, and it can be very challenging. If you think about some of the experiments you've been involved in, we're going to go over one today by Dr. Thomas that was extremely complicated, and talk about how we used a protocol schematic to make that easily understandable by the reviewers. If people are reading something and have something to read in front of them, you can only imagine how complicated it would be if you have a 5-minute presentation to talk about your experiment and you have to talk about all the different controls you used. It would be a lot easier if you could just use a picture. So, and then the last thing we'll do is review assignment number 3. Okay, don't need to underline that. Just some quick housekeeping before we get going here with our protocol schematics. Big thank you to everybody who's either emailed me, stopped by my office, flagged me down on the road, something to let me know things in class that could be better. You'll notice that we have our different lectures now composed in the module unit, not just listed in the announcements. What that's going to do is it's going to allow you to hopefully see what you need in each of your for each of your lectures a little bit more easily. So for today, you'll see the lecture video, which we're recording right now. You'll see a PDF or a Word document that is the notes. So if you'd like to take some notes along with this, you can download it beforehand. You will also see in the example PowerPoint file that has the animated protocol schematic. And that's just going to be for your reference if you want to use it for the future to kind of see how that works. And then you'll have the assignment and also the discussion link right there. So those 4 or 5 things will be right in that module titled Protocol Schematics. You won't have to go to different places, and you won't have to kind of think about what is needed. With that said, though, if more things come up as we're going through this class, please make sure to email me. As I said, this is my first online class that I'm teaching, and I want to know before the end of the semester things that could be better. Because if I can make them better right now, I'm gonna, and that's gonna make our experience in this class a little bit better. Okay. We also got some exciting news this week. We did get approval for the headshots, so you'll be seeing a kind of a doodle poll or some other availability poll for us. We're going to be taking headshots over at the co-library one morning in a couple months, hopefully when it's a little bit warmer out, but it will be inside. And a local professional photographer is going to be taking those headshots for us, and those are what we're going to use in our lecture where we talk about creating a LinkedIn profile, creating a ResearchGate profile, and possibly a Google Scholar profile. I find this very helpful for people, especially if they're going to be in the job market soon. Okay, that's the housekeeping for today, so let's move on. Protocol schematic number 1. If we want to define what a protocol schematic is, all we're going to say is that it's just a diagram, and it's going to give us a quick little snapshot of what the study is. Normally it's going to be less than 1 page. You do not want to see this being a very long and drawn out schematic. Typically, for some of my studies, I might have multiple. Some are kind of way zoomed out on the overall protocol of the study. And then let's say we have an experimental day that gets repeated. We might have a zoomed in one that's only on what happens on that experimental day. So for example, Here's what a good protocol schematic looks like right here. If we zoom in on this guy, right there, we can see this is for an experiment that had 2 different days, okay? And we can see that we have time on our x-axis right here. And what this is going to do is it's going to differentiate between day 1 and day 2. What is the timeline look like, and what experiments or what measurements happen during that time? And we can see, I have not read this study, but I can tell from this schematic that there are muscle biopsies occurring. There are also meal ingestions that are going to occur. And if this is a glycogen depletion and dietary manipulation day, I'm going to guess that this is a low-carb diet that they're going to eat at this time point. Then, about 2.5 hours later at 10 a.m., they're going to start their exercise. This is a 4-hour exercise protocol. Then, after that exercise protocol, at 2, 3, 4, 5:30, and 7, we're going to have some different amounts of meal ingestion. So I'm going to assume this is kind of some small split up amounts of meal. And then at 11, your clock, you're going to have a meal ingestion as well. Okay, this is going to be high or low carbohydrate. And then we have 1 muscle biopsy on that day. So from this schematic, we can tell exactly what's happening in day 1. If we look at day 2, this is our experimental test day. This is a day we have our meal ingestion, we have insertion of femoral catheters, and rest for a long time. So this is going to enable us to get a slow infusion, probably of some marker molecule that they're going to use when they take repeated muscle biopsies right before they start exercise, in the middle of the exercise, and then at the end of exercise. I've done a study like this before where I had to have a muscle biopsy and then run for 70 minutes. Luckily, they used a lot of lidocaine, so it wasn't too bad, but I can tell you it was not super comfortable the next day. Here we go. Here's the variable intravenous glucose infusion, and that's what they're going to be looking for in these. With this, you can also see that they're doing breath and blood sampling following the femoral venous blood flow. Okay, here we go. These are the time points for each of those, and we can see that we 1, 2, 3, 4, 5, 6, 7 of those. And what this will help us do is when they talk about maybe some statistical differences in the results section, they'll say that the immediate pre was different than the the third to last sample. By referencing this schematic and those results together, you can kind of get a better picture of where those measurements took place in the overall experiment. And that's what we're trying to accomplish with this protocol schematic. Okay, so that's what a good one would look like. Let's zoom out here and let me show you an actual example of one that I recently received. Let's see if I can do this. Yeah, and we'll zoom out just a little bit more. Here is a protocol schematic that I actually received as a part of a recent article to review. And I looked at this and I said, this needs to go back to the authors. They need to redo this. What this has is 2 different time points here. You can see that they have different study days here, which is okay. That's fine. Maybe we would want to do that on a study timeline, a little bit more like this instead. Then we have our urine sampling, and it gives us the type of urine sampling that was happening, morning and 24-hour collection on days 1 through 4, and then you have every void collected here. I don't know if that's actually true, but from reading it, I remember that. And then my favorite part right here is this last row, diet protocol. Yes, 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 yes. This gives me no information. All the authors would have to say is that the dietary protocol was practiced throughout the entirety of the data collection, and that would give me the exact same information that this line does. So table format, not going to be great for your protocol schematics, but looking at this one up here where we have some overall timing, that's number 1. We want to make sure we have that. We have some measurements that are happening, when those measurements are happening, and then also a little bit of evidence of what type of experiment this was. Because we have that right here. We see high or low carbohydrate diet, and then we have variable intravenous glucose infusion. That's going to tell us that this is probably a matched or crossover type study. Okay, so let's move on a little bit. So we've covered kind of what is a protocol schematic overall and some examples of some good ones and some bad ones. Now let's move on to why we need these. So here's an example from my dissertation right here. And this is not necessarily a protocol schematic, but this is a schematic that is more related to what I was looking at in my dissertation. And in that, we were looking to impart some exercise stress, and then the person was exercising at a certain intensity. This was either prescribed by heart rate or by rating of perceived exertion. And that person would then appraise that exercise stress, the intensity of this. And here's the two ways that they would appraise it. Either, am I at my target heart rate? If we told them to be at 150, they could look down at their heart rate watch during their running and say, oh, I am at 150, okay? And then they would respond. They would either increase, decrease, or maintain. So if they were prescribed 150 beats per minute and they looked at their watch and saw that they were at 150, they would theoretically be more likely to maintain than a person who was using their rating of perceived exertion. So this schematic right here gives us an overall kind of psychological evaluation. I'm sure that if any of you have taken a class with Dr. Reddy before, he has used a fair number of these to describe some of the models that he has used. Here's an example of a not great one that I used during my overall defense. And we were looking at the type of running. It was 3 to 4 sessions a week, 60, 80 minutes per session, and a variation from 150 to 270 minutes per week of running. And we said, okay, on their days, they would be fit with their heart rate monitor first, then they would do a warm-up, then they would have the workout described to them and have their exercise intensity assigned based on this big giant RPE chart that I made, and then they do the run. Okay, doesn't give you a lot of information, but I wanted to give you a picture of kind of where I was back in the day when I started making these and where we've kind of moved since then, and how we can get better. So let's zoom out. So the first reason that you would have these is for your thesis or grant proposal. Okay, number one, that was the thesis right there. So if any of you are thinking about doing a master's thesis, now might be a good time to start thinking about how to delve into designing a protocol schematic. And for your assignment, if you've already started making one for your master's thesis, I'm not going to have you make a new one. You don't have to go find a new article. Use the one that you're doing. Just try and make it better and describe how you've made it better based on today's lecture. We'll get to that at the very end. Okay. Another reason that you might use one is for institutional review boards, especially if you're working with human subjects. You're going to want to have to describe what your experiment is. Here's an experiment that we did that, I said tables aren't the best. Well, sometimes if you use a table in combination with a different format, it can be good. And what we're doing here is we're trying to give a schematic of here are our 5 different groups that we're recruiting. And what this is doing right here is it's telling us that on days 1 through 3, when all these people are in the experiments, they're all going to get 1 liter per day. And then on day 4, we have a change in the amount of fluid they get. Our control group stays at 1. Our group B gets extra water, half a liter. C gets an extra 1 liter. D gets an extra 1.5. And E gets an extra 2.25 liters. Okay? Then if we combine that with this figure right down here, this tells us a little bit more about their daily experience. This is once again that zoomed out version, and now we're zooming in on a typical day in the experiment. Okay? The participant will wake up, they will drink their water with breakfast, then right after breakfast at around 9 o'clock, they'll provide a spot urine sample. Okay? Then they will provide another spot urine sample right before lunch. They'll drink some more of their water, provide another spot urine sample after lunch. They'll go until before dinner. Repeat that again. Here we have our evening window, and then we have our evening spot and our last water right before bed. And you can see that on day 4, which corresponds to this up here, when they get their extra water, here we go. XXX mLs, that tells us we're drinking our extra water at that time point. Here we have again, here we have it again. And what this does overall is it shares with the Institutional Review Board that you have a consistent way of distributing the water and also of taking your measurements throughout the study. This is going to give them the nice warm fuzzies as they look at your experiment, and it will allow you to then send this on to your research group. Which brings us to our next one, grants. And for some grants, we have required graphic outline. Here we have the World Health Organization and some of their grants right now. Recommended format for a research protocol. They require a graphic outline of the study design and procedures using a flow diagram must be provided. They require this. If you're going to be considered for this grant from the WHO, this should include the timing of assessments, okay? You need to have that figure. And the reason that you need to have that is because you're going to have people from a bunch of different backgrounds reviewing your grant. They want to, as an organization, understand that all of the people reviewing this grant are going to have a good idea of what you're doing in your protocol. So that's why they require it. They don't require it just to, weed people out. They want to make sure that we're getting a quality evaluation of everybody's grant, and therefore they require it. So if you're going to do a grant, you're going to want to have one of these as well. And typically, if you do one for a grant, guess what? You can probably use that same figure for your IRB and in your thesis proposal, defense, what have you. You're going to use these figures a lot, so I will encourage you to take a good amount of time to make them as good as possible. Another area you can use this is during data collection. There's two examples that I'm going to give here for data collection. Here's a first one where we talk about men and women being brought into a study. And what this is going to be good for is to post in your lab for the people who are helping to collect data. We talked about this when we were going through standard operating procedures as well, the importance of having these. Well, we also want to have these for everybody, maybe in a graphical format if it's something that's a little bit easier. So this is kind of like a graphical SOP that also can be called a protocol schematic. We can see here with our women who are going to be on this side, or women and men. This side over here, we're talking about urine collection. And then over here, we're talking about whole blood collection. We can say we go EDTA and serum, and then the order of the measurements overall. Then we store them, okay? And then we'll do some metabolic profiling later. Once again, this is a zoomed out version of what your analysis is going to look like. If we wanted to give an example of a zoomed in, here's one once again from my dissertation. And let's zoom out just so you can see the whole thing right here. For my dissertation, we had a lot of different analyses that we're going to be doing. Do not do what I did here. Add shadow. That is dumb. It makes me angry just seeing it right now. Let's remove that right now, if I can. Font. Oh, it's not letting me do it. Oh, because it's a picture that I copied from PowerPoint. Ugh. Anyways, don't add shadow. What this did is this told us that during our fasting blood draw, I was going to be taking 1, 2, 3, 4, 5 different tubes of blood. Depending on the analyte that we wanted to do for this, we were going to be using a different tube and how much volume we needed for each of those. For example, our first one right here is our CPT tube, which is our cell preparation tube. This we use to evaluate the expression of GLUT4 transporters in the mononuclear cells here, the PBMCs, in that blood because they were going to be doing some run training. We wanted to see if their GLUT4 was upregulated specifically. We looked at 1 and 3 as well. And this told all the people in this study how many times they were supposed to invert that. So if I was doing the blood draws and I had a person helping me do the blood draws, I would give the tube to them, and they would know right from this that, okay, CPT tube, I'm going to invert that bad boy 8 to 10 times. After I do that, we have Dr. Lee, Jenna, Colleen, and Dylan, who were people who were helping me out, that we would deliver that tube to. Okay, they had a separate SOP, and I'm sure Josh can fill anybody in on what to do with a CPT tube after you get it. Okay, that wasn't part of what we were doing for our experiment. Okay. Second part, okay, we had 2 4 ml EDTAs, and this was going to be for glucose and insulin concentration. What we did with those, invert them 8 times, centrifuge them for 15 minutes, and then it told you after you centrifuge them for 15 minutes, and it also has a little reminder to use the refrigerated centrifuge, you're going to aliquot them. Then you're going to get 6 total microcentrifuge tubes, and you're going to place them in a freezer and how to even label. And the last 2 are going to be the ones that go to Quest Diagnostics. So we have a 4 ml EDTA for hemoglobin A1C, and also our serum separator tube was used for our lipid panel. These we inverted either 5 or 8 times. One got left at room temp, and then this one had to be within 30 minutes. And you have a form that you fill out with those so that Quest Diagnostics could pick them up later in the day and do the analysis for us. So here's an example of a protocol schematic that is more similar to an SOP for what we're going to be doing. So an example of how we can do that. Let's move on. We've done our grants. We've done our data collection. We've done our institutional review boards, and we've done our thesis presentations. Next one is going to be publications. Okay, so here's an example of what they call a subject recruitment tree. And if you're using human subjects in your research studies, this is something that is becoming more and more required by certain journals. There are some journals out there that will not accept your paper for review unless you have a figure that looks like this. Okay? What this does is it tells us the number of subjects that were enrolled, how many were excluded, why they were excluded. After that, we have 44 people right here who were allocated either to the control group or the intervention group. And then from there, we follow up, and then we have, okay, what about that? After exclusion, we have 1 excluded here and the reason that they were excluded. 2 excluded here, 1 because they had to travel, and 1 because they had dizziness associated with the project. And the last one tells us how many were included in the analysis. This is going to typically give more detail than what you have in your written methods. What this does is it allows the reader and the reviewers to say, this person did a good job and they kept track of all their subjects. It's not just a random number that we're throwing out there of the people that were included. We know how many people were enrolled, we know how many people were excluded, and the reasons for why they were excluded. If you're working with an IRB that keeps track of this, or if you have a registered clinical trial, you need to have this information. You will have your study shut down if you do not provide this on your yearly review. So it's important to keep track of this. So what we're going to do now is we're going to make one based off of one of my older protocols. So let's read this through real quick here. We had 2 parts to an experiment. All right. Part 1, 125 healthy men and women between the ages of 18 and 45 years participated in the current study. Of these, 10 were excluded for the following reasons. Failure to follow water restriction protocol, that's what WR stands for. Failure to provide sufficient spot samples. At least 4 of 6 samples had to be produced each day. We have 8 people there. And then failure to return the final dietary log. Okay, and it references figure 1 right here. Part 2 of this protocol schematic is going to be, on day 4 of the experimental week, participants were randomly assigned into one of the 5 groups of the graded rehydration intervention. We went through that before when I showed you right up here. Here's the 5 groups that we're talking about. Here's day 4. They get 5 different amounts of water. Okay, so if we go back down to here, maintaining 1,000, that's their control group, or increase based on the amount. Okay, so we have extra half liter, extra liter, extra 1.5, and extra 2.25. So let's go over and see how we would make one of these. I know this isn't too complicated, and for a lot of you guys, this is going to be kind of a little bit of a review. But what I want to do is show an example right here of what this looks like overall. Here we go. And if you look at the start, in the text, we said 125 people participated in this study. And that is true. 125 people did participate in the study. But we have more information that's important to share. Let's look up at the top. If you see here, we had 637 people who responded to the initial call for subjects. So we sent out a bunch of emails, and you can talk to my past graduate student, Ainsley and Hillary, about how many emails they tried to get these subjects. And they found that from those 637, 495 did not meet the initial inclusion criteria. So what this study required is that we reach out to 637 people, get in touch with them, go through the initial criteria, and we determined from that most of them did not meet the criteria. And we have the rationale in a spreadsheet somewhere of why they didn't meet that criteria. Then from there, you move on. Okay, so if we subtract 495 from 637, we get 142. That's the number of people that provided consent. Okay? Then we randomized them, okay? That's when we gave them which group they were going to be in down the road. From there, we had people who voluntarily withdrew prior to completing the participation. If we subtract 17 from 142, we get that 125 people completed the participation in this 4-day study that we showed above, the 3 days of the decreased water and the 1 day of increased water. Well, we wanted to have 25 in each of our groups. We already gave the rationale why people were excluded after the fact, but now what this figure does is it tells us a little bit more which groups had people excluded and for what reason, okay? And then we know from this that we have 115 people included in the final analysis. So we can see from this figure overall, we started with 637 people. How did we get to the 115? So if a person was to look into the paper and see the degrees of freedom on one of our statistical tests were different between the groups or the comparisons, they would be able to theoretically go to this figure and figure out, oh, that's because they had 3 people excluded from the G plus 50 group. I get it. That's why it's different when we compare these two groups from one side to the other. Okay? So I want to now pull up a version of this that I've just changed a little bit and go through some of the things that are important when we're making these. I would hope that in PowerPoint right now, everyone knows how to make a text box. And I'm going to actually post a nice kind of how-to that Alec Muthig from IT developed for us last time we produced this class. Because last time we did this class, we did it in person. Half of the class was a lecture. Then the second half of the class, we went over to the computer lab and actually did this. The difference that we're doing here is the lecture is online, and then you do the work on your own. So you'll find a little how-to document with this week's module as well, so that you can use it to make this. If we want to make some of those figures that we were talking about before, if we look at up here, we have kind of 2 arrows that are happening. So how we do that is we're going to insert a shape. And we're going to go to our different types of arrows. And we want our arrow with the elbow. And what you'll notice is that when you go to your text boxes here, you'll see some little dots. We want to click on our first dot there. And now you have the availability to change this figure or this shape. Where do we want to put it? Well, we want to put it right to that nice little dot that you saw pop up on our figure there. There we go. So we have that one. Now, if you look down here, a default on PowerPoint is this dumb blue. I hate it. If you see people presenting and they have the colors as the default colors, I immediately think, well, this person didn't try very hard. So, and when you submit for publication, you have to make sure that everything is in black and white. Otherwise, you're going to get charged for having color figures. You don't want that. Your mentor doesn't want that. The readers don't want that because it's not what they're used to. So what are we going to do here? We're going to format this, format our shape right there. And here we go. What do we want to do? We want to make it a little bit thicker overall. We'll bring it up to 1.5, and then we'll change our color to black. There we go. If we wanted to do a couple other things, we could change what type of connector we have there at the end. Typically, you're going to want to use this one right here. You can also, if you want, you can add a begin arrow type on there as well. Right now you can see it's blank, but if you wanted, we could have an arrow there. That would be indicative of something that's flowing both directions. We don't have that flowing in both directions here, so we are going to remove that beginning. Okay, there we go. But now we also want to make sure that we say, okay, well, some of these 637 are going to our 142. So let's add in, insert, once again, a shape, and instead of using the elbow this time, now we're just using a straight one. And we're going from here right to there. Okay? And same thing with this. We are going to make sure that we have the same thickness, also that we have the same color overall, and now you can see that's like that. If we were right over here, you would see that you have the ability to either make it straight up and down or over here. Looking at this, we can use this and select our next text box. If we're actually connected to that middle dot, that arrow will move with us as we go. And we want to make sure that we have a nice perpendicular. There we go. So that's our part one that I wanted to show you about how to format those arrows, because that's important. The next part has to do with spacing, okay? One thing that you'll see when you move these is you get all these little arrows and kind of liners pop up. These are important for kind of how things look. And I am kind of a stickler for how these go, because I think it's important to have things look good to the reviewers. So if we look right here, you can see that that red arrow with the double heads right to the right of my cursor right now is the same size as the one that's below to the right of the randomization arrow. What that's doing is telling me that the space between my n equals 142 and my n equals 637 is the exact same distance as between my randomization block and my n equals 125 block. That's something that I want, okay? If I was going to be a real stickler, then I'd want to kind of look at this one as well and make this one the same too, okay? Now, if we take this, we can bring this one in, and what I would want to do with this is make it line up with this one before, because this is giving us some information about what's happening during this first phase. So as you can see, as I move this to the right and to the left, we get different liners. Right there is going to be in line with my n equals 17 box. I want it right there. And then the next thing I want is I want it to be equidistant vertically between my n equals 637 and n equals 142. If you look, you can see those little orange tabs to the right. That tells me that I'm equidistant. Right there, they're gone. I'm still aligned with my n equals 17, but I'm not equal distance between the two. Move it up. There we go. Now, let's get a little bit more complicated when we go down here. If we have our 5 different groups here, we have to be very careful when we're aligning these to see when we get the same spacing between each one. And it looks like right about there. There's those two. There's that one. And you can see that there's a little bit of a discrepancy there. What I'm going to want to do is kind of look right down here. And I want to align it right with that. And then go up from there. And that should give us some good alignment overall. As you can see, this is going to take a fair amount of finagling, a lot of detail orientation, okay? It's important to do this. If you send this in, the example that I steal from my buddy Matt at the University of Arkansas, he always gives the example of a car oil change. You know, I'm sure a lot of you change your own oil out there, but for me, you know, East Coast guy, I'm not changing my own oil. I take it over to, you know, the grease monkey, and I get them to do it for me. And every time you go back into your car after you get your oil changed, they have that nice little paper mat sitting right there on your floorboards under your accelerator and everything. And that gives you a really, you know, kind of good feeling overall. You think, oh man, these people took care of my car. Does that mean that they changed the oil any differently as somebody who didn't put that little mat there? No. But what it does is it makes you feel that these people care about what they're doing. If you present a figure, a protocol schematic that is all wonky and things are not lined up, just like this, okay, is that telling me any different information? No. The information in my boxes is exactly the same. But what that does tell the reviewer, anybody who's seen your experiment for the first time, is this person doesn't care enough to line these things up correctly, to make it look nice. Then the question comes in, well, are they going to care enough to properly sterilize their glassware before they do their experiment? Probably, but I'm a little bit less convinced than I would be if things were designed correctly, you know? And, you know, there's a lot of different ways to do this, but I've found that having a good protocol schematic is a great way to build some confidence with people, okay? So the last thing we're going to do here is we're going to insert those kind of double bend curves here that we saw. If we look up here, you can see we have double bend. One bend there and one bend there. So how are we going to do this one? Let's go back. Once again, insert our shapes. We'll go to our elbow, find that middle point. Okay, now we will go out and find this middle point. If we want to adjust where that lies, we can click on this little orange dot to say, oh, I want that a little bit lower, or I want that a little higher. We're going to kind of put it right in the middle, or leave it at default. There we go. Now, we're going to do something a little bit differently with this one, because formatting takes time. I like to save time. Let's do it all right now. We said we were at 1.5. There we go. I'm going to just copy this. And I need 1, 2, 3, 4, 5 of them total. But I'm actually going to just make 3 copies. 1, 2, 3. Okay? There's number 2. There's number 3. There's number 4. And you can see that we have a nice lined up figure. How am I going to get to this one? Insert shape. Let's just use a regular one for this guy right here. Once again, find that middle point. Oop, not that one. Click on the thing that you want to move. Find that point. Change it to 1.5. Change our color back to black. There we go. The other thing people don't really worry about too much is the joint type. I like to use the miter joint, and I also like to use a square kind of end. That gives you a little bit more definition in some of your figures overall. Okay? So with that, we've been able to show you, you get your text boxes, you make them similar size overall, and then we add the arrows to give us, okay, what's our general flow and what happened along the line. Make sure that you do this. This is something that's really important. If you're doing your proposal or your defense for your thesis, you're going to want to be able to tell your audience what happened to all those subjects that were recruited, okay? In order to get our subject number, how many do we have to screen? This gives us extra credibility, because if we had certain exclusionary criteria for this protocol, what this is doing is it's telling everybody, yeah, they really went through it. They screened as many people as they had. Look at this. They had like a 20% acceptance rate into this study overall. That means they did a lot of work to make sure. I'm more confident now that they actually used their exclusionary criteria and just didn't say that we excluded for something. If I know they excluded 495 people from this experiment, okay? That might be something important for an animal study. Maybe animals, as they tend to, may pass away before they were supposed to for the study, and you have to exclude them. Well, you might want to get credit for working and doing the intervention with all those. I'm sure that that's a lot of work associated with that, and that might not be the norm for your journal, but it would be good for your defense to say, hey, look at all the work I did. Okay? Add that in. So there is our subject recruitment figure. Let's move on to one of my favorites. This is when I was a young assistant professor, first semester, coming in here, taking over for those of you who may have remembered Dr. Paul Thomas, who's a fantastic scientist who does a lot of cardiovascular phys. He's an emeritus professor in our department currently, and he'll come in and do some guest lectures for me when I do my cardiovascular section and exercise phys. He had a very complicated protocol. I am not going to read through this methodology section right here. You can do that on your own time if you'd like, but this is what he had in one of his papers. It was a paper with data that he had collected from 1980, and in my first semester he came to me and said, hey, Evan, I got these reviews back from the reviewers. They like the paper, but they don't understand my protocol, okay? And I read through his methodologies. Yeah, it keeps going here all the way down to there. And I said, yeah, that's a complicated methodology. Let me read this and see if I can break it down. So let's go through this. They had 3 phases to this protocol overall. And these phases were exercise testing, kind of baseline testing. And then what they did is they used a medication that depressed heart rate, either propranolol or atenolol, which are beta blockers, and they use those to depress heart rate, and then did exercise testing with these 3 different types of beta blockers, or 3 different types of heart rate depressors being used in each person did all 3 of these conditions. Well, they wanted to make sure that they had the correct dosing for the medication beforehand, so they had to do a really complicated protocol. So after their first visit, they gave them some days of recovery, and then they had their second visit where they did a max workload determination, where they wanted to find out, okay, what's 65% of your cycling efficiency. Then on the first day of their medication titration, you can see here they cycled at 65% of their max, and then they took their medication for 3 days before their visit. Well, they took either 80 milligrams of propranolol or 50 milligrams of atenolol, and then they came back for visit 4 a couple days later, and we increased the dosage by 60. Okay? Well, there's 2 outcomes to this second dosage. Well, first, if their heart rate decreased less than 5 beats per minute, so while they're exercising at the 65% of their max, was the heart rate, their max heart rate, less than 5 beats per minute different between these two? If it was less than 5 beats per minute difference, then we can say that there's no added benefit from adding on more medication. Therefore, this person is going to get the lowest dose possible. But for some people in this, adding extra medication actually caused their heart rate to go down more. If that was the case, well, then they came back for another visit, and they got even more of each medication, and you had the same outcome. Well, did their heart rate not change from the previous visit, or did it decrease by more than 5? Well, if that was the case, they came back for a final 6th visit to really max out their dosage here. And then for all of these people, we had a heart rate decrease. And from this figure right here, you can see that if in all these cases, then they moved on to the counterbalanced experimental part, where they did maximal voluntary contractions, 30% of their max, and they sustained it for these 3 different types of exercise. They did that each under 3 different conditions, which was washed out over 7 days. Whew. Complicated protocol. But I know for myself, I would much rather look at this than read that and be able to tell the difference. Or if I'm going to read that, be able to compare it and contrast it with a figure to really give myself a good grip on what is happening. So, after Dr. Thomas came to me, had me develop this figure, we submitted it with our round of revisions, accepted, like that. Okay? So this was something that the reviewers wanted to see, and we'll go into working with reviewers a little bit later in the semester, and now it was accepted for publication. Okay? Are we going on to our last one? Yes! Here we are. Hang with us, guys. Got about 15 minutes left here in the old lecture. Last one is presentations. Presentations are super important as you progress in your career, because this is how you're going to get the information about your science out to the population. And it's really important that everybody understands what you did during your experiment, especially before you go into your results. If they don't understand your experiment, they're not going to be able to understand or put context to your results. So I'm going to give an example right now of one that I did and presented at a conference a long time ago during my postdoc. And this was a complicated protocol because we had a screening section here. We also had an intervention phase, okay, where we did something different for our groups. And then we had an oral glucose tolerance test where we took some measurements, okay? So let me pull up the figure. Okay, here we are with our PowerPoint. And what I'm going to do now is I'm going to try, can I adjust this? Yes. I'm going to adjust my screen briefly here. Oh. I guess I can't increase the size of it. Let's see if I can do slideshow. There we go. And what I'm going to do is I'm going to kind of move my camera around while I'm going here to show you what's happening. Okay? So first things first, we have our method slide. Now, oh, I I guess while we're recording, I have come to find out that you can't record animations. Ah. Maybe I can do this. Yes, I can. Okay, here we go. So, if we have our figure right here that you guys are watching, you can see that what I'm doing now is an animation. We had our screening. They would meet with a registered nurse and determine if their hemoglobin A1C was greater than 5.6%, which means they probably have type 2 diabetes. Then we move on to our intervention phase, which was 3 days in length. We had one group during this intervention phase that was euhydrated. We had another group that was hypohydrated. Okay, or actually the people completed it in two different conditions. The euhydrated group got 3 liters of water a day. When they were hypohydrated, they got 1 liter a day for day -3, 1 liter a day for -2, and half a liter for day -1. Then we had our oral glucose tolerance test. Let's move this guy over here now so you can see this one. During our oral glucose tolerance test, we did a blood draw at the 0-minute time point. Then, after they did that blood draw, they consumed 75 grams of glucose. Then we had blood draws at 15, 30, 45, 60, 90, and 120 minutes. This figure shows you how those all align. And we used animation to help guide the viewer through all those phases. So if we go all the way back to the beginning, we had very little up there. And this allowed us to kind of use this as more of a script to take them along with us. We didn't put the whole figure up there to start with and then go from there because that would have been too much information. Okay, let's go back. Everybody's back with me. So let's talk about how we did this. If we look at this figure overall, we are going to add in our animation pane. If you look over here to the right-hand side, you can see just how many animations were needed in order to get this to be correct. Every single item on here had to be animated separately. So, with things that appear, we have to have appearing. With some things, we have to have disappearing, okay? With some of our lines, for example, let's look at our first example when we had our animation from screening to meet with an RN come in. Well, with that one, besides having just that straight connector come in, that shape, well then we had to look at the effect options. Once we pull up our effect options, that tells us what direction does this want to come in. From the bottom, from the left, from the right, or from the top. Okay, and we had this one from the bottom up to the top because that shows us the direction. During the screening, they did this. Then we saw that our meet with our RN actually changed after. So if we look down a little bit further, text box 53, we know that that's eventually going to change. First, it appears, and then a little bit later, it's going to change. Let's see if I can find it. I guess not. If we look at our effect options overall, oh, here we go. After the animation, look, we have something in here to change the color. After that animation was complete, it's going to change color, which is going to dim it a little bit. So therefore, here's the colors that you can dim to. It's going to take it out of consideration. So if you don't have to pay attention to that something afterwards, you can just remove it. Another trick I like to use a lot with these is adding in some gray bars. So here's a gray bar. That's the same background. And what that enables us to do is, okay, well, we have our full table here that we're going to add in, table 56, that was added in. But when I first add that in, I only want to talk about my euhydrated group. So if I only want to talk about my euhydrated group, I'm going to have that table 56 appear at the same time as some other things, okay? As the same time as this box. And then I can remove that box right here. And when we remove that box, then it goes away, just like that. One thing you want to be cautious of when you're doing this is the ordering. Overall, if you look right here, you can look at bring to front. If I send this to the back, right there, it goes behind my table, and now I can't see it anymore, okay? It doesn't help me out. It's not going to block that out if it's in the back. So I want to, once again, bring it to the front, and there it goes. Now we can use that to block off the sections of this table that I don't necessarily want to use or describe while I'm talking at this point. The reason this is so important is because, like I said, it assists you with your audience to give them examples of how you want them to follow along with your story. When you give a presentation, you're telling a story. If I just put up one of my protocol schematics, so for example, let's say I put this up just right there on the screen while I'm talking and said, okay, we did this thing. If I just put that up there, it's too much information. You're going to take your entire audience's attention off of you, and now they're just going to be staring at this figure for the next 5 minutes trying to figure out, okay, visit 1, all right, well, I did this, and then there's 2 days, okay, well, blah, 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 what about the medication titration? But if you start with just that exercise testing box, and then go to visit 1, and then go to this right here, and then go to here, that's what I want you to be able to do. So part of your exercise, I'm actually going to add it into our assignments tonight. You can either create your own original. We might as well move down to the protocol schematic assignment, which was 75 points, due next Thursday. You can either choose your own, or on contrary, what I'm going to have you do is I'm going to post this as a PowerPoint slide with all of these parts already in there. What I'm going to then have you do, I might eliminate a couple to make it a little bit of work for some of these arrows overall. Then I want you to animate this, okay? Give it to me one piece at a time, okay? When you do this, maybe you do both of these arrows together, but I want to see this animated in a way that you would use during a presentation, okay? That's one option for what you can do. The other option is make a new one. So let's say you do have a thesis presentation or a proposal coming up, and you're like, man, it would be really helpful if I had something and could get credit for it. Well, here's your opportunity to do that, okay? What I want you to do is either use an article. So if there's something published out there that's using the same protocol that you're using, or you might not be doing a research protocol, you can choose one from a different article. What I'm going to say here is don't choose a paper that already has a protocol schematic. I'm going to look them up. If you just repeat what's in there, that's not really getting any benefit for you for this protocol. So find a methodology that doesn't have a protocol schematic along with it. Then I want you to include that research methodology or describe the experiment just in a few lines, just so I know what we're working with. Then tell us the purpose. What's the purpose of that? That's going to define kind of the level of detail you go into with your protocol schematic. Is this for general population? Are you giving this presentation at the local YMCA for general population? Then I'm not going to really go into some of the depth of the controls that I'm using. Or is this something for my mentor that they're going to know if I'm doing things right or not? I want that extremely detailed then. Do you have some good photos in there? As we showed before, we had just these little photos of the syringes that tell me about blood draw. That's really nice to add in here as opposed to having words. When we look at protocol schematics, we want them to look good. So adding some appropriate pictures here and there is going to be good. If we go back to my one from my dissertation, here's an example of some other good pictures, because these show us the actual color of the blood tubes that we're going to be using that help with our kind of standard operating procedure there. Are they correct? Okay, moving on. Is there a clear flow from the beginning to the end? Do I see where we're starting and where we're ending? Then I want you to be creative. I've gone over some very basics of protocol schematic design in this lecture today. I want you to use your own creativity, and I know you're thinking, I went into science because I'm not creative. or not a creative person, but I want you to dig deep. Ask your friend, ask your grandma, hey grandma, how can I make this look a lot cooler? Hopefully they'll give you an idea of adding in something. But what I want you to do with this is alert me to it. Sometimes people add in things I've had in the past where they add in a really cool design, but I don't see it and they don't get the credit. Make sure you give me a little heads up of to what the creative thing you did was. I want those text additions to be appropriate and clear and concise. Do not have long paragraphs in there. The whole reason we're making these protocols schematics in the first place is to avoid that wall of text. We don't want that. And last, this is the most important part for me overall. That's why we have 20 points devoted to it right here, is that a reflective paragraph. What have you kind of learned from this lesson overall? How are you going to apply this in your future? I want to see how what we've gone over today has really helped you kind of either evaluate for the first time or reevaluate what you knew about protocol schematics. And I'm just hoping that I've stressed the importance of these. This is kind of one of those things that a good protocol schematic is not going to get your paper accepted into New England Journal of Medicine. Sorry. But a bad protocol schematic is something that can get your protocol schematic rejected in the first place. Okay, so a good one's not going to make up for bad science, but a bad one is going to be one more check in that negative column when it comes to their master editor's kind of decision of either to accept or return it back to you for some major revisions. So I think this is an easy place where you can pick up some good brownie points with reviewers. Also a great way that you can instill some confidence in your audience if you're giving a presentation or you're submitting a grant. So with that, good luck on the assignment. Check out for the discussion points as well. I'm going to give a couple examples I'm going to ask for some feedback on in the discussion section, and we'll go from there. I'll have grades posted later this week or early next week for our first assignment, and we'll go from there. All right, guys. See you next week.